Right, in this recording, we want to take it to the next level of eukaryotic transcriptional control and talk about what factors are involved in controlling the initiation of transcription, literally loading the RNA polymerase on to get an mRNA transcript of a DNA gene. Again, differential gene expression is what we're after here. It's kind of the key to molecular biology. What genes need to be turned on at what times to accomplish what we need for that living thing to keep on living. It's also then the source of many genetic diseases where we have differential gene expression that has gone awry, and cancer is kind of the prime example there. If we can understand this better, then we can understand the disease better. If we understand the disease better, maybe we can even cure it. So we've talked about access to the DNA and talked about chromatin modifications, acetylation of histone proteins. We talked about methylation as a way to shut down a gene, methylation patterns, and epigenetics. And now, once we have access to the DNA, how do we actually say this gene needs to have RNA polymerase come down and read it and make an mRNA transcript? Most people, when they're talking about expression of a gene, are referring to this event, transcriptional control. Now, ultimately, in many cases, you don't see an effect on a cell unless the protein is active. But most people will still refer to expression of a gene just at the transcriptional control level. So keep that in mind as you go on and read more in molecular biology. So again, we talked about chromatin modifying enzymes provide an initial control, but then how do we bind the transcriptional machinery to make sure that we make an mRNA? So we're going to talk about control elements in eukaryotic genes. So again, these are parts of the DNA, parts of the gene in essence, that are non-coding but are going to help to regulate transcription. And we can have both negative and positive regulators. We can have repressors, anything that downregulates or shuts down a gene, and activators, anything that upregulates or turns on a gene. And it's much, much more complicated than we see in prokaryotes. There are many, many, many more players. Now, because of that, we're going to have a much simpler explanation, and let's go through that now. So what we see here is levels of control. Again, so here's our model of a piece of DNA with a gene on it, and I want you to focus on the promoter. We see our promoter, and then we see downstream of our promoter to the right, which that would be 5' to 3', are exon and the introns of the gene itself. Now, just upstream, or just 5' to the promoter, we see what are called proximal control elements, near control elements. So these are sequences of DNA that are going to bind to proteins that are going to affect how the promoter functions, how it loads the RNA polymerase on to transcribe this gene. Now you'll also see a little break in the DNA, that double hash mark, which indicates a large distance, not proportional, that are distal control elements, far upstream. These can be tens, if not hundreds of thousands of base pairs away from the gene that they are affecting. And that's always been a very big mystery in biology. It's how are these things that are so physically separated from each other, how do they affect genes so far away? And we'll look at that here in just a second. Now, what we talk about when it comes to gene expression in eukaryotes are transcription factors. Now, we've already mentioned general transcription factors when we talked about transcription just in a general sense. So we have transcription factors that are involved in binding RNA polymerase in both prokaryotes and eukaryotes. And so when we remember that, we talked about the ta-ta box, transcription factors come sit down, and then we load RNA polymerase II on. So that was the Shiningdale-Garneau sequence in prokaryotes, and there's a simpler system, but there's still transcription factors that are necessary for RNA polymerase to work in prokaryotes. But where we have an operator oftentimes that binds a repressor or not, or we have an activating protein in prokaryotes, here we have a whole slew of factors that are going to be specific transcription factors, specific to a gene or a set of genes that are related, but may not necessarily be anywhere near each other in the genome. All right, so let me say that one more time. These genes may not be anywhere near each other in the genome, and that's in direct contrast then to the operon model. Right? So let's take a look at that as we go. So again, we're going to talk about proximal control elements that are located close to a promoter. These seem to make sense. And distal control elements, which we use a fancy word called enhancers. And you'll hear about a technique if you go on to molecular biology called an enhancer trap. And that's a way to locate sequences of DNA anywhere in the genome that affect a specific gene expression pattern. So we'll talk about activator proteins that can bind to enhancers and stimulate transcription of a gene. Okay, so this is upregulation with enhancers. So if we look at a model of how this works, we can see there is our promoter, there's our Tata box, and there's our gene. Okay, the simplified version. Way down far away, we have our distal control elements and our enhancers. Activators, proteins that recognize the DNA sequence of the enhancer, bind to it. And then one explanation for this is that we have the DNA actually bend to bring the distal control elements, the enhancers, near to the Tata box and the promoter. This is how they then engage this group of mediator proteins, other specific transcription factors, with the general transcription factors to ultimately help load RNA polymerase on and let it run. So again, this is so vastly simplified. If you look at the number of little blobs they've shown you in this picture, you can really see that there's a ton of stuff going on. And it's fairly well understood, but at the same time, every additional factor you add gives you an additional level of resolution for control, a way to change the system to fine-tune your up or down, or to turn it off or turn it on. And we're going to see that's really, really important when it comes to eukaryotic gene expression. All right. So again, some transcription factors are going to act as repressors, and some are going to act by influencing the chromatin structure as well. So we're going to go back to that idea of access to the gene as initially the first thing that has to be controlled. And what's doing that? Other proteins. So they're going to be called repressors or promoters. All right. Now, combinatorial gene activation is the idea that I've got genes that have to work together. More specifically, I have protein products that need to work together. So again, that was the operon model that we had in a prokaryotic cell. You just literally put all the genes next to each other in front of one on and off switch. And when you needed them all, boom, you threw them on. It's not so simple, nor is it so often the case where it's all or nothing in a eukaryotic system. It's much more often the case that you may need two genes in many cases, but not the third one, or a fourth one only in a specific case. So you have to have a finer degree of control over individual genes, but still be able to activate all the genes needed in a certain situation. So if we take differential gene expression with respect to development, we can look at the different promoters and activators that we see in a liver cell versus the lens cell that you see in an eye. So in the liver cell, we're going to look at the activation of the albumin gene, and in the lens cell, we're going to look at the activation of the crystalline gene, which is actually one of the proteins that makes the lens of your eye work. And the key is this. You see upstream of each of those at the top of the diagram here, you see the diagram of both genes. Both of these cells have both genes, right? Because every one of your cells has all your genes. But you'll see upstream of both promoters of the genes are different sets of enhancers. Both of them have the gray enhancer, but the albumin gene has the yellow and the red enhancer in addition, while the crystalline gene has the orange and the pink enhancer in addition. So what we're going to see is that in the liver cell has gotten signals to turn on genes that produce the yellow, the gray, and the red activators. Those then will turn on albumin gene. The gray activator is expressed in the liver cell, but it binds to the enhancers for the crystalline gene, but I need all three to turn on the crystalline gene. So I don't want the crystalline gene on because I didn't turn on the orange and the pink one. All right? So this is kind of a catch-22 because I've got to turn on genes to create the activators that will then in turn turn on the gene of interest that we're talking about. And so we'll talk a little bit more about that as we talk about development. But continuing with this example, we took a look at the lens cell now. So in the lens cell, we're looking for the orange and the pink and the gray. And so again, in the lens cell, we express those three enhancers, which bind to DNA and turn on the crystalline gene, Well, then again, the gray, which overlaps between both genes, enhancer is bound, but because the red and the yellow aren't there, we don't turn on the albumin gene. All right? So this is the idea that if I flood the nucleus with a specific recipe of activators, of proteins that are going to bind to enhancers or proximal control elements, then all the genes that fit that recipe will turn on simultaneously. They do not have to be located next to each other. They can be scattered all over the nucleus, right? And so we then have a greater degree of combinations that we can make by looking at the different elements that I need to turn on a specific gene. All right? We'll talk about this more as we talk about development and gene expression in general.